Science of the Seasons is environmental monitoring citizen science program that Esperanza and some of the others here on the call of us started back in 2010. We started organizing for this. We're monitoring species in addition to rockweed, which is our focus today, but we have a total of 19 species. And we have a wonderful group of partners throughout the state and actually nationally who, like the Phenology Network, help us with our data, folks in New Hampshire Sea Grant, and UNH, as well as Acadia National Park, the Schoodic Institute, the Maine Botanical Gardens, Maine Audubon, and of course, Maine Maritime Academy.
Sorry, Beth, I'm so sorry to interrupt. Folks, you have to mute. You have to mute your phone, and you have to mute your little microphone icon that's on your GoToMeeting control panel. We are recording this, so many other people who wanted to be on this today can hear this. So it would be really great if you could. I see 3 people that need to mute themselves, either on their phone or on their the computer little green microphone icon. Thank you. Sorry, Beth.
No problem. Thanks, Esperanza. And also the U.S. Fish and Wildlife Service. And Jessie will be sharing some great information about her research. I just wanted to orient everybody to what we're doing in the program and how volunteers are involved. So as some of you probably know already, but phenology is not a common word. It's the study of the timing of plant and animal life cycle events. So everything from migration to leaf out and flowering of plants, as well as reproductive activity and metamorphosis. You see the monarch butterfly chrysalis there, as well as lilac flowers and American toad. And then, of course, the picture on the bottom left are receptacles, reproductive receptacles from the Ascophyllum nodosum plant, which is commonly called rockweed. And these are all things that are easy to notice and record, but a big impetus behind this new effort globally, really, of tracking and monitoring phenology to help with climate change research was during the Intergovernmental Panel meetings on climate change in 2007, phenology was formally identified as the simplest process in which to track changes in the ecology of species in response to climate change. These changes are happening all around us all the time, and we can see robins building nests, we can see trees leafing out, and by recording and monitoring those very local scale changes, we can help ground truth climate models and make them really more useful at the local scale. And so each of our different species has different characteristics, phenology phenophases they're called, changes that we're observing. And for rockweed, because it's not a plant or an animal really, we have different procedures for monitoring rockweed than for our other species. And the information, as Jessie will share, is going straight into her research. But I'll just run through what it is we're doing so those of you who are not involved can have a better sense. So we're looking at the phenophase changes, which I'll share a bit more about in a moment. We're collecting temperature and salinity in the areas where we're doing our monitoring. And then we're also collecting age and growth measurements of individuals within rockweed stands or at our sites. And so rockweed, until I got involved in this, I wasn't closely observing these changes myself, but since then, of course, I've become much more attuned to these changes over time, and it really, through an annual cycle, you see drastic changes in these reproductive receptacles, what they look like, and whether or not they're even present. And so you see across the top in our phenophase observations, on the upper left, you see where there are new side growth in the areas where the receptacles will grow, and then they change from being smooth and flat to inflated and then sort of dotted and what we call dotted inflated, which is rough. And then once they've released their gametes, they're torn and eventually they drop off. And then apologies for the side view here. This is a card that we print out for our volunteers to use in the field. But rockweed, similar to the way trees have growth rings, they form air bladders on the main stems each year. And so you can actually count those to get a rough estimation of age and measure the distance between them from year to year to understand the growth rate of any given stand of rockweed on average. And then here's, this is, I won't bother going through all of this, but this is a card that we hand out to help our volunteers who are monitoring water temperature, because the reproductive changes are cued by water changes, rising water temperature in the spring. And then we also collect salinity data to understand how freshwater inputs and other aspects from one location to another are changing with the water salinity. Our volunteers collect their data in the field and then use our website. This is a screenshot of our website. There are different data sheets where they go in and enter their data, and then Jessie uses it to inform her research. And so that's just a quick overview. Our website is there at the top of the page, and then this is a couple of photos, some of you who are on the call today, of our wonderful volunteers. And then our names, myself and Esperanza as co-coordinator, Sarah Randall, who's on the call, who's our assistant coordinator, and Christina Rivka, who's our summer intern currently with Science of the Seasons. And so with that, I will turn it back over to Esperanza and Jessie.
Thanks, Beth. I just want to quickly introduce again Jessie Muhlin, Dr. Jessie Muhlin. She's an advisor to our program. She's been really instrumental in our development of our protocol, our resources to volunteer observers, data management and analysis, and so forth. So we're really happy to have you today, Jessie.
Great. Well, thank you very much. And it's really a pleasure to talk about rockweed and have everyone who is on the call now, as well as people who might view this in the future, to have interest in rockweed and the Maine Science of the Seasons program, because it really is a great way to get involved and collect data that can be used to inform some really interesting science, or I think so. Let's see. So I thought I would give a brief introduction in terms of what rockweed is, because it's a common name that actually encompasses a number of different seaweeds. So rockweeds kind of encompass all of what we call fucoid macroalgae. So these are seaweeds that are intertidal, and they're lithophytes, meaning that they're rock loving. They need a hard substrate to attach to and to grow. So if you go to sandy beaches, you wouldn't really find rockweeds because that substrate is moving too much, and that those individuals can't attach and then grow up over time. Within the Gulf of Maine and kind of the North Atlantic, there are two dominant rockweeds. The first one is Fucus vesiculosus, which is much shorter in stature and is often found in more areas of higher water motion. And the species that most people think about when they hear rockweed is Ascophyllum nodosum. It's often much larger in stature and is found in more sheltered embayments. It has a variety of actually a lot of common names, including Norwegian kelp, knotted rack, knobbed rack, asco, and egg rack. And so you can kind of see that there are some similarities and differences in terms of looking at Fucus versus Ascophyllum. And so one of the first things is just to kind of acquaint yourself with, when talking about rockweed, are you talking about Fucus or are you talking about Ascophyllum? And that both have air bladders, and when they're reproductive, they both have receptacles, and I'll kind of delineate those two for you in a little bit. So it's always very helpful to have a common vocabulary when talking about seaweeds. Most people don't learn about the algae collectively, and so we like to kind of give analogies to other organisms, what we might know more about or have learned about. So oftentimes seaweeds are, the analogy is to plants. And so I want to just kind of clear up some perceived misconceptions that rockweed is not a plant, so it doesn't have a root system or a vascular system. It is photosynthetic, but if I use the word plant, I don't really mean it in terms of the word plant. I mean as an individual. And so rockweed attaches to a hard substrate. And at the very bottom of this diagram, you'll see that where the holdfast is. So it attaches by a holdfast, and then it starts to grow. And it grows apically, meaning that it grows from the tip up. And so you can kind of see on the upper left-hand corner a little arrow that points to the apical tip of the frond. And so that's where the newest growth is always going to be. Rockweed has these air bladders that after about 2 years of age, they start to lay down an air bladder for every new growing season. And so you can actually count those air bladders to get a relative age for each individual. And then another thing that often is confused with an air bladder is the reproductive organ, which is called a receptacle. And those are, when they're present on the individuals, are found laterally across the frond or the thallus. And if you look in the lower right-hand panel of pictures, you can see a kind of a blue circle that's identifying those receptacles, and then a little line that's kind of trying to indicate a cross-section. So if you took a receptacle and you had a little razor blade and you could kind of make a dice cross-section of it, what you would see underneath the microscope, either a dissecting microscope or a compound light microscope, would be inside that receptacle, these cross-sections that you see on that at the left-hand panel of that picture. Males have these flask-shaped, and females do too, of these conceptacles inside the receptacles. And for males, they have packets of sperm that are stored in structures called antheridia, and females have packets of eggs that are stored in structures called oogonia. And they look different underneath the microscope. Males are bright orange in terms of their antheridia. The sperm have a carotenoid pigment, so it senses light. And so if you were able to look at it, it would really look like a very bright, bright orange. And females are an olive green in coloration. Ascophyllum in general has a tremendous amount of structure, both at low tide and at the flood tide and at high tide. And so I hope that these two photographs kind of emphasize that the structure is present no matter what the tide is doing, in that a lot of the mid-intertidal zone is covered quite extensively with Ascophyllum individuals. And so at low tide, they kind of lie on the intertidal substrate, and they provide a tremendous amount of habitat and desiccation tolerance for lots of other organisms that are living on or beneath them during the low tide. And as the tide comes in, the air bladders of the Ascophyllum individuals allow it to become much more buoyant. And so that at flood tide and high tide, you can see that there's a tremendous amount of three-dimensional structure that Ascophyllum provides in the nearshore environment. And so because of its dominance in the mid-intertidal zone and also the amount of physical structure it presents in the intertidal zone, rockweeds are really ecologically important. They're perennial species, so they live for many years. They're foundational, meaning that once you have an Ascophyllum individual that establishes itself and a population grows up over time, it changes the physical and chemical parameters of that intertidal zone and allows for a lot more biota to be present and thrive, as well as Ascophyllum is a primary producer. So it's fixing carbon dioxide, it is releasing oxygen as a byproduct of photosynthesis. When it gets detached or deceases and it goes through the detrital pathway, it's really important in terms of nutrient cycling. one thing to kind of keep in mind in terms of thinking about Ascophyllum is that there is a tremendous amount of variability of Ascophyllum across the coastal populations. And so what Ascophyllum looks like in southern Maine can be very different with what Ascophyllum looks like in terms of its structure in, let's say, Cobscook Bay. And so there is lots of variability, and one of the wonderful things about the Signs of the Seasons is that observers are able to kind of observe all those differences and be able to record those data and share them, and so that we're able to get a really nice, large spatial understanding of all of that variability. And the other thing to kind of keep in mind is that the natural world is really not static, and so the timing of reproduction for Ascophyllum is really a great tool to be able to see that things are really dynamic, and that this species really responds to changes in the physical environment and the chemical environment, and that it's really a wonderful indicator species for changes in climate. So one example in terms of just thinking about the variability of different populations is just looking at receptacle size at 3 different locations in 2014. There's even among the individuals at each of those sites at Schoodic and in Machias and at Harpswell, you can see that the coloration, the amount of epiphytes or other algae that are growing on top of it are all really different. So the location in which Ascophyllum persists is really this amazing interface of a tremendous amount of physical and chemical and biological forces. And so I thought I'd kind of entertain the idea of how dynamic that intertidal zone is in terms of the physical and chemical forces. And if you look at this, it's kind of, you can think of it as either the tide going out or the tide coming in, and that band of rockweed is being exposed. And at a high tide, the water is going to be completely submerging those seaweeds, and at low tide, they're going to be completely exposed. And so if you think about all those physical and chemical forces, there's changes in light quality and light quantity as the water comes up over the individuals. There's changes in nutrients that are available for photosynthesis and other metabolic pathways. There's lots of temperature stress. The intertidal zone experiences hot summers and really cold winters, as well as desiccation in terms of being exposed or immersed in water. Osmotic shock in terms of the differences between salt water and fresh water. So you can imagine that individuals at low tide during rainstorms like the one we might have had today really change the water dynamics that might be available to the seaweeds, as well as mechanical damage. So the force of waves and the drag that each individual has as the wave is pulling it back, as well as in the Gulf of Maine, we have lots of ice during the winter, and so ice scour is also another mechanism for mechanical damage that these seaweeds have to endure. As well as not just chemical and physical factors, but biological factors as well. So most common in terms of thinking about the ecology within rocky intertidal habitats is competition for space, so overgrowth from other algae, and then predation in terms of herbivory. So lots of intertidal fauna like snails and limpets that like to graze and eat small Ascophyllum zygotes or babies, as well as graze on or upon the adult or juvenile individuals. One thing to also keep in mind, although I would prefer that we don't really talk about the implications for it, is that Ascophyllum is a commercially harvested seaweed in Maine. It's been harvested for centuries in the North Atlantic, and that it has a lot of wonderful characteristics and qualities that are used as a marine resource. So my specific interest in research with fucoid algae is looking at reproduction. And so we know that rockweed reproduction is influenced by a number of physical and chemical factors, and that rockweeds in general share these reproductive ecological systems with other marine organisms, so including corals and urchins, in which all of these species have external fertilization, meaning that they release their eggs and their sperm into the water column, and reproduction takes place external from an individual. Depending on the species, a species can either have just be male or just be female, or it can be hermaphroditic, meaning that it has both male and female reproductive components. And for Ascophyllum, it is what we call a dioecious individual. It has separate male and female individuals. And so you can see on the figure on the left, that is a female. It's got these olive green oogonia that are releasing from those conceptical pores. And that you then have these bright orange antheridia for the males on the right. And so if it's releasing, you can see the difference between males and females. And generally speaking, the sex ratio within any particular population is 50-50. The amount of reproduction that Ascophyllum has is quite amazing, I think. So this is just an example of the amount of reproduction that any one individual might have, or we call reproductive allocation. This does vary both in space and in time. So for this example, in 2014, I had my student Sarah Bruchou, who is in this photograph here, collect 5 male and 5 female individuals. And then I had her take all of the receptacles off each individual and count them. And then we dried both the receptacles and the vegetative matter, the things that were not reproductive, of every single individual to get the percent of the overall biomass, dry biomass, that was allocated towards reproduction. And you can see in terms of thinking about mean number of receptacles on the graph, that's the panel on the left, that, that there are hundreds, if not thousands, of receptacles for every single individual. And so every single receptacle has multiple conceptacles within it, and within every single conceptacle, there are heaps and heaps of antheridia that have many, many millions of sperm or eggs. So the amount of reproductive output is really tremendous for this particular species. And then thinking about the reproductive allocation, just looking at dry weight, nearly 20% of the overall biomass during the reproductive season is allocated towards reproduction. So that is a tremendous amount of energy that is towards the effort of reproducing for the next generation. And so that, because I'm so interested in the reproductive ecology, the phenology is also really important. So not just how many receptacles, but when those receptacles are present, and the timing of that maturation of the receptacles and when eggs and sperm are released is of real importance. And I'll hopefully allow you to see kind of the implications or the repercussions of that reproductive initiation and gamete release. And so what we know is that the timing is associated with photoperiod, so the amount of daylight, and also sea surface temperature, so the water temperature. And in general, although it will also vary in time and in space, the maturation time and takes place in the winter and early spring, and that the gamete release occurs in April and May. In June, the receptacles start to get torn and they are shed, and then the new growth and kind of initiation takes place in July and through the autumn. And so because rockweed is known to entrain its reproductive ecology with photoperiod, but also most importantly with sea surface temperature, changes in climate can really influence changes in the reproduction of these rockweeds. So the seminal paper that is the foundation of this research was by Bacon and Vadas in 1991. And what they did also with sites in Maine was that they saw that there was a span of time between 3 and 7 weeks in the spring that Ascophyllum was reproductive, and that through very detailed sampling, that 6 degrees Celsius was the onset of gamete release. 10 degrees Celsius was at the midpoint of the percent of gamete release that took place with 50%. And that at 15 degrees Celsius, it was really the termination of gamete release. And so that really became the baseline for a lot of the phenological data that I started to acquire and have help from Maine Signs of the Seasons Ascophyllum observers. The other thing to kind of keep note is that if water temperature is the primary indicator in terms of when reproduction occurs, that within the Gulf of Maine, we're really seeing a significant trend of increasing sea surface temperatures. And so this graph shows you long-term temperature trends for one buoy within the Gulf of Maine in the the New England Ocean Observing System buoy set. And what you can see, and hopefully that you identify, is that there is a trend from when the data started to be acquired until now, that the sea surface temperature is increasing over time, and that within the last 12 years, there has really been a marked increase in terms of much warmer temperatures experienced in the Gulf of Maine. And so the idea is that with increasing sea surface temperatures, there could be shifts in the reproductive timing of Ascophyllum. And that one of the research questions I have is that how important is surplus eggs and sperm and zygotes to the nearshore environment? Again, it's externally released. So I kind of think of eggs and sperm and fertilized eggs, which are zygotes, being really important nutrients or particles for other intertidal organisms that might rely on them as a nutrient source, energy source over time. And that if indeed they are important, that if you have shifts in climate that produce shifts in reproductive timing, that there could be a trophic implication for that. So that if you have a snail that is dependent on Ascophyllum reproducing in the spring, and that snail is over time adapted to be ready to acquire that material in, let's say, May, and the sea surface temperatures are warming so that Ascophyllum is shifting its reproductive timing to, let's say, April, those kind of trophic implications might bear out. So this is one research project that Maine Signs of the Seasons has really helped in terms of increasing and supplementing some of the data that I'm trying to collect to see shifts in reproductive timing over a number of different locations over a number of different years. And so one thing that we did starting in 2014 was to put out temperature data loggers. So these were deployed in the winter and early spring and could record the temperature every 15 minutes. And the idea was we could then compare the temperature and then the state of the release of gametes with the Bacon and Vadas 1991 literature and see is there a similarity or difference in terms of timing. When the Bacon and Vadas research was going on, they didn't have fancy data loggers. They just used a stem thermometer at high tide, and they took an instantaneous measurement so that these data loggers provide more precision in terms of thinking about the temperature within the field. And then what we also did was refine kind of what the gametes were doing inside the receptacles. And so we created an index that kind of allowed us to assess what the gametes were doing in terms of maturation of eggs and sperm and then release of those Antheridia and Eugonia over time. And we also used the Signs of the Seasons data to kind of also see the changes over time. So the Signs of the Seasons data isn't as invasive in terms of doing cross sections and looking underneath the microscope, but provides a tremendous amount of overall phenophase differences at each different location and over a number of different years. In addition, we completed a number of field studies over 2 years to identify the intertidal animals that might utilize that surplus reproductive material from both Ascophyllum and also Fucus vesiculosus. So we had 3 sites across the coast that we sampled extensively in the spring, the summer, and the fall. And what we were trying to do was be able to collect all of the macroscopic invertebrates, and then using a technique called stable isotopes, resolve food web dynamics and be able to see what might be eating different things in the intertidal zone and whether or not Ascophyllum reproductive material is an important component of any of those intertidal organisms' diet. We also did a number of small laboratory experiments using seed mussels up at the Down East Institute on Beals Island. And so these are just some figures looking at the release of eggs and sperm artificially, and then feeding those to itty bitty little mussel babies in the laboratory. And so the data are really interesting in that we're seeing that there are patterns of variability. And so this is a chock-full slide that shows you on the left-hand side 2 sites that Signs of the Seasons observers collected over 3 years, where you can see that over the course of time, so on the top axis, you have the months, so April, May, June, July, and August, the primary time when Ascophyllum is reproductive. And then what those phenophases are the different colored bars that you can see. And so what you can see is that there's a lot of variability. And so in thinking about changes in climate, you really need to have a number of years to be able to look at trends. And we're getting there in terms of having more than one location and more than just 1 year to be able to then see patterns. And also you can see on the right-hand panels the original Bacon and Vadas data showing kind of the onset, the midpoint, and the termination of gamete release. And then the data that we collected showing the onset, the midpoint, and the termination of gamete release. And so the data are not exact, but they really help fill in an understanding in terms of how temperature really does influence the reproductive timing. And so the hope is that in time, if there are continued changes in sea surface temperature, that we would see this reflected in the appearance and the duration of Ascophyllum phenophases. And it can really be a wonderful indicator in terms of seeing changes over time. And then also thinking about the differences that might exist across the trophic food web of organisms that might rely on the reproductive material that Ascophyllum provides. And then the second research project that includes Signs of the Seasons volunteers is a recent project that was started this past fall. And it's looking at Ascophyllum population structure in coastal Maine. This project, the lead scientist is Susan Brawley at the University of Maine, and includes a number of researchers across the state, and also includes one researcher in Atlantic Canada. And the idea for this is to be able to understand long-term field site data that looks at both sheltered and exposed locations, that those locations have either experienced harvest, high levels of mechanical damage in terms of wave force, and both include rocky intertidal zones and soft sediment habitats. And this is also to help kind of see that variability and understand that variability of growth rates and age structure and density of individuals at each of those locations. And so the Signs of the Seasons data also helps in terms of thinking about that protocol C, in terms of thinking about the age and the growth rates of populations that Signs of the Seasons observers might also be collecting. And so I selected a site and started a site that's in Harpswell, Maine. And most of the other researchers use students in their courses. And I tried using citizen scientists, tapping into some of our Ascophyllum observers to help in the field. And I would really say that Signs of the Seasons volunteers are fabulous field research assistants. They have knowledge of the Ascophyllum, they have interest, they are already really acquainted with that habitat, and they are, I would say, much better than the undergraduates who might be required to come to a lab and collect data. That the Signs of the Seasons observers are wonderful about it. So if you are an observer or interested in observing, or you've listened to this webinar and you're interested in participating in this research, please let Esperanza or Beth know. We're going to be sampling again in the fall, probably in October. We would love to have other citizen scientists come out into the field and help us collect the data, because it's going to be a long-term project. Preliminarily, in terms of thinking about the data that we are acquiring, there were 3 sites that were baseline information was collected over this past fall. And so they were at Pemaquid Point, at Harpswell, and on Sears Island. And what you can see is that, again, there is similarities and differences in terms of the length of the Ascophyllum individuals and the age of the Ascophyllum that's found there. And so we're very excited in terms of continuing this research and collecting more data to see trends over time. So with that, I'd be happy to answer any questions, and I really thank you for your attention.
Thank you, Jessie. That was incredible. That was really great and very informative. So you can type your questions into the chat box on your panel there. So I had a question going back to, while we're waiting for people to type their questions in, to the research, the first research project. And can you go back a few slides, Jessie, to, wait a minute, stop. So what did you note about, this is the slide, the animals that, are you able to detect the bivalve larvae? Are you able to identify them in your samples, or?
No, in terms of the bivalve larvae, no. So in the work up at the Down East Institute, those blue mussels were about 1 to 2 millimeters in size, and they were seed mussels that were cultivated there. So we didn't collect them in the field and then take them back.
Right. Okay, great. Thank you. So Susan Domici has asked regarding research project number 2, does the soft sediment habitat support Ascophyllum growth?
That's a good question. So I would say that soft sediments in general don't support Ascophyllum establishment or growth, but within soft sediment areas, there's often kind of cobbles or boulders or kind of eroding softer substrate that Ascophyllum can attach to and grow over time. The differences in the growth may not have to do with the soft sediment, but in terms of thinking about water motion. And so often softer substrate locations have lower water flow, and that might encourage higher growth rate versus a location that might be more wave exposed and might have slower growth rates over time.
And Beth is asking you, could you describe how the box and whisker plots in your last slide are showing differences in variability?
Sure. So they are showing the the darker line is the mean, and then the whiskers are showing I think the 25% confidence intervals. And what you can see is that if you were comparing Pemaquid Point and Harpswell, there really isn't any difference in terms of the mean Ascophyllum length. And Sears Island has a tremendous amount of variability in terms of the Ascophyllum length. And the mean length is greater, but not statistically different than Harpswell or Pemaquid. And an interesting thing to kind of keep in mind is that Pemaquid Point is a high energy environment. Sears Island is a low energy environment. Harpswell is a low energy environment, but experiences harvest. And so what you can see is that those lengths might be reflected in terms of harvest might be very similar to natural energy that's experienced in higher wave motion locations like Pemaquid Point.
Great. And can you tell us how many years of data you'd like to have to start to be able to see more clear trends in the timing of reproductive onset in the spring?
Sure. I would say that for most climatic data sets, between more than 5 years, between 5 and 10 years would be very helpful. So we only have, well, after this season we'll have 4 years, but 4 years doesn't show you that there could still be a lot of interannual variability. And so that you're not able to really see patterns over time. You're just seeing the noise of the variability that might exist naturally without changes in temperature. And so the longer the data set continues, the more evidence there will be if there are patterns of shifts of those phenophases earlier compared to later in the season.
I have another question too. We'll take George's question, Guilfoyle. He said he lost sound. Well, in audio, yes, we'll make that available. Question about the slide that you're on. So the Bacon and Vadas 1991 data versus your data 2014, 2015 to 2015. Mm-hmm. It looks like. So maybe a little more explanation. So at the mid-level, mid-stage, that's quite a big difference.
Yeah, in terms of 7 degrees versus 9.7 degrees.
Yeah, that's a huge difference because at 10, it's at the higher stage. So I'm not sure. A lot of it has to do with there were different locations that were chosen. That the difference between taking instantaneous temperature versus taking temperature that's from data loggers, that I'm really able to have much more confidence in terms of that's the temperature that was experienced during high tide across the entire reproductive season and beforehand. And so I think that there could be a number of reasons why there is a difference. Mm-hmm. And also methods, perhaps?
Exactly, the methods.
Mm-hmm. Yeah. There's another question by Michael Adams. What time of year would we expect to see the new side branches begin to form?
Yeah, so the new side, it depends on the location. So different locations are going to have kind of different trends in their growth. But when the receptacles are shed, they leave those little side branches or little side laterals that then start to have kind of new growth.
And we saw a lot of variability in that on sites. So how long for each species to grow to full harvest after being cut? George?
Yeah, so that's another good question, George. And a lot of it has to do with the locality. So, depending on each location, there are some locations in the Gulf of Maine that have higher growth rates than others. So, it really has to do with growth rate over time. And so it would be hard to have a definitive time. It really would depend on the location.
Have there been any studies that show that that growth, or is it all new?
Well, you would be able to see in terms of measuring growth rates from each air bladder to see what the growth rates were. And I don't know of any comprehensive research that has looked at that from lots of different locations.
Mm-hmm. Well, hopefully in 5 to 10 years we'll be able to
exactly
support that data. And George is also asking, are changes in water temperature and acidity, et cetera, affecting growth of these species?
I don't see why it would not. All of those factors play a role in terms of the growth of any individual. And so in terms of thinking about water temperature, metabolically, there's going to be kinetic changes when you increase or decrease water temperature. And in terms of thinking about ocean acidification, for a photosynthetic organism, that's a carbon question of, is there going to be more carbon dioxide or carbon speciation in the water that's going to be available for photosynthesis or not? And so, yes, indeed, it's definitely going to affect the growth of Ascophyllum and other fucoid species.
Great. Are there any other questions?
These are all great questions.
So, okay, we have a couple more, or one more. Since Ascophyllum is not a plant, what should we call it when we're talking to you? And what should we call it when we're talking to a newspaper?
Good question. I know, it's so hard. I would say that to call it an individual or a seaweed or an alga, because it isn't a plant. I mean, it's evolutionarily very distant from a plant. So I would try to use individual or seaweed or alga.
Yeah, good one.
But those are words like seaweed people know, but alga is a hard one. That's not really in people's common language vocabulary data set.
True. Beth, I don't know if you asked. Oh, okay. How many years would you ideally like to continue this research? And are there other research questions you'd like to look into in the future?
Yeah, I would say that many, many years. A lot of research questions have this time span of kind of a funding cycle. In terms of like maybe 3 to 5 years, and that really doesn't allow you to see real changes over time. I think one of the wonderful things about some of these research questions is that we can utilize the expertise and interest of citizen scientists to extend that time and the space across the Gulf of Maine that people can collect data. And so ideally, like decades worth. I mean, it would be wonderful if people just made looking at the phenology of Ascophyllum a ritual in the spring and in the summer, and that they teach their children and their grandchildren, and they continue to do it. I mean, that would be the ideal. But I'll keep doing it as long as I'm around because I think it's really very valuable, and it provides a lot of insight in terms of changes across the nearshore environment that might not be captured with other organisms that might be offshore, or terrestrial organisms that people might be seeing and hearing about in the news.
Mm-hmm. So,
and then as for, yeah?
No, go ahead.
And as for other research questions, there's always lots of other research questions. And so one potential research question that colleagues and I are pursuing is looking at not just the relationship between Ascophyllum and fucus reproductive material to marine invertebrates, but also the link to birds. And so those coastal birds that are very important as well, and kind of seeing the food web dynamics between fucoid algae and macroinvertebrates in the intertidal and coastal birds that are of interest.
Yeah, that's really important. The question that we get a lot from our volunteer observers is, once the gametes are released and there's torn receptacles, is there a reason to continue monitoring or making observations and collecting water quality data throughout the summer after that?
It depends. I mean, I think that if once those receptacles are torn and they're shed, then just kind of the onset of when new receptacles are present is helpful to record that initiation of new receptacles. And that if people aren't observing, they may miss that. Mm-hmm. And I think also if you keep observing, you might be observing things that others might not be observing. And so the more you get to know the population that a person is observing, they might see things that are occurring there that they can document. And so I think having sustained observation is very helpful.
Mm-hmm. Great, thank you. Any other questions? Well, seeing none, Jesse, want to thank you so much for this invaluable research that you're doing and your connection to Signs of the Seasons, and thanks to all of our volunteers out there. Again, this is the first of several webinars that we're going to be doing over the summer, so stay tuned to the Signs of the Seasons website. And if you could hear us, we're applauding. And thanks again, Jesse.
Okay, thanks so much.